Okay, class, continuing with male reproductive physiology. This is where we left off. Okay, let's see what we have here. These are seminiferous tubules, which you see in the blow-up drawing here. These are gametogenic cells, which will make the sperm and so forth. This big cell here is the Sertoli cell, sustentacular, also called nurse cell. Now, the point I want to bring out right now is the Sertoli cell does a lot of stuff. Okay, it does secrete inhibin, which we talked about, that inhibits FSH. It does secrete the MIF, malaria inhibiting factor, which turns off the development of the female reproductive anatomy, so we can become males. And it does secrete something called androgen binding globulin, which concentrates the testosterone and androgens, the testosterone, within the tubule to assist in the development of the gametes, which would be basically the sperm. All right. But the issue I want to come with now is this. Once you study meiosis, you will see in meiosis you have something called crossing over of the genetic material. Crossing over means that part of mom's chromosome will be on dad and part of dad's will be on mom. That occurs in meiosis. So when you look at the genetic material of the sperm, there's a combination of that. So it's not totally, the sperm is not totally male. Okay, I mean, as far as from the male, shall I say? Okay, I mean, there's some components of the mother's DNA, the man's mother's DNA, and so forth. In addition to that, if you recall, in immunology, early along in utero, you develop something called tolerance. Tolerance means you tolerate your own cells. But there were certain areas that you did not develop tolerance to because it was not seen. For example, the blood-brain barrier. Well, since sperm are being produced constantly, you would have not developed tolerance to them either. So bottom line, sperm is foreign to the male. It's in the male, but it's foreign to the male. Now, what does that mean? The immune system would attack it. Okay? So we have to protect the sperm from the immune system. Okay, so one of the things the Sertoli cells do is to protect that by forming what we'll call the blood-testes barrier, sort of like the blood-brain barrier. Another thing that the Sertoli cells are, they'll produce an FAS ligand. A ligand is a generic term for anything that can bond to a receptor. This FAS ligand then will turn on the T lymphocytes that try to get there and make them undergo apoptosis. Remember, programmed cell death. The FAS receptors in a cell, if they are turned on, cause the cell to self-destruct. Okay, so that's one thing. The other thing is the sperm do not really have the MHC1 and MHC2s. Major histocompatibility 1 proteins or major histoglycoproteins or major histocompatibility 2. So all of this is in an effort to try to protect the sperm from not being attacked by the male's immune system. Okay. The other thing is, as you're developing the spermatozoa, you're going to get rid of a lot of the cytoplasm in the developing sperm cell, and the Sertoli cell phagocytizes that extra cytoplasm, and that extra cytoplasm is put into something called residual bodies. Okay. So spermiogenesis is when you go from, let me go back here, when you go from the spermatids to the spermatozoa. And all that's happening in the seminiferous tubule. Okay? All that's happening in the seminiferous tubule. And I described that earlier. I need to develop the flagella. I need to put an acrosome. I need to get rid of some of the cytoplasm in the spermatid. And so those are factors that must be done in order to develop into the spermatozoa. Okay? And the genetic material must be called titer, and that's where the histone proteins are somewhat replaced by protamines, and the protamines condense the genetic material, so it's called titer into this little head of the sperm. The sperm, of course, spermatozoa would have a head, midpiece, tail as it develops. Now, once you develop this, then you would be in pretty much at the lumen, and once you're at the lumen, then these will float away and ultimately go into the epididymis. First in the straight tubules, then the reedy testes, then the efferent duct tube. The efferent duct tube is in the epididymis. Okay? Alright, so we'll go a little further. Spermiogenesis, maturation of the spermatids and functioning spermatozoa. Protamines replace the histones. That causes compaction of the chromatin. Nucleus changes. Flagellum develops. Also the acrosome, which will be placed on the top, will also develop. Cytoplasm removed by Sertoli. Acrosome, a cap of digestive enzymes. That cap, those digestive enzymes, are the ones that's got to cut away at the corona radiata around the ovulated egg, ovum, and also the zona pellucida, which surrounds it. The mitochondria are located in the midpiece, and of course the mitochondria are important for what? For the whipping action of the flagellum. Okay. We talked about the testosterone in the brain earlier. That's the enzymes. Okay, we'll go. We talked about this at age 70. Okay. So I think I made it. All right. We mentioned that 1,000 to 2,000 germ cells would move from the yolk sac into the testes. We discussed that in meiosis. So again, I'm coming down. Spermiogenesis. Cytoplasm removed. Here again, that's the Sertoli cell there. Here the residual bodies, which are extrusion of cytoplasm from the developing spermatozoa to get rid of that and compact this thing. So these residual bodies are being, as you can see, digested by the Sertoli cells. Now the sperm, once they leave the Sertoli cells, which would be up in here, are non-motile. Motility will not occur until it really does more, it kind of matures more in the epididymis, but actually motility will not occur too much further down that track there. Okay. So we'll go a little further here. 59. Okay. Here again is a picture of the acrosome, which will digest, as you can see, try to digest that corona radiata, which we haven't talked about yet. That's the female, and the zona pellucida. That's off the top of the head. It has enzymes. Midpiece, that's the midpiece, which has the mitochondria, and here's, of course, the tail, and so forth. Okay, go down some here. Sperm development requires Sertoli cells, create a blood testes barrier, which we talked about. The fast ligand, which we also discussed there, to do the apoptosis that would occur of the T cells so that they don't attack. Phagocytize those residual bodies, we spoke of that. The Sertoli cells also produces androgen binding globulin. The androgen binding globulin, again, is important to concentrate the testosterone into the tubule for sperm development and so forth. Okay. Okay. So testosterone or its derivative is required to stimulate meiosis in early spermatid. Testosterone, again, just to remind you, is produced by the interstitial cells. It's which are also called Leydig cells. That is stimulated by LH, luteinizing hormone. FSH enhances spermatogenesis, but actually what you really need is this testosterone binded to the androgen binding protein. FSH ensures optimal fertility. Now what that means is testosterone is the primary hormone to do spermatogenesis. FSH is needed and it gives an optimal plan to do it, but is not completely essential. But without the testosterone, the androgen, it would not happen at all. So FSH is an adjunctive chemical to go along with the testosterone. Okay. Now we already have looked at the anatomy, so we already should know that spermatids move from the seminiferous reedy testes, and actually would be spermatozoans. But spermatozoans move from the seminiferous tubules, reedy periductus, epididymis, and then the epididymis is where we talked about the maturation. They become more motile, but not completely motile there in the epididymis. And the epididymis, again, is where you store the sperm. It's not stored in the testes. It's not stored any place else. It's stored there. And ejaculation is spermatozoa move from the epididymis, ductus, ejaculatory duct, and the urethra. Seminal vesicle and prostate gland add fluid. The seminal vesicle adds 70% of the fluid to the semen, and the prostate gland around 30%. The seminal fluid contains fructose, which is your energy source, and the prostate contains citric acid, calcium, coagulation proteins. The coagulation protein is generally called vesiculase. And remember, the coagulation is important when the person ejaculates. That particular semen should stick on the vaginal sidewall, and then fibrinolysin will dissolve the vesiculase, which is the clotting chemical, so that then the sperm can go motile, and then the prostaglandins that was secreted also from the seminal vesicle will cause the mucus in the cervix to dissolve so the sperm can get up, and the prostaglandins will also cause a retrograde action in the uterus to propel the sperm up into the body of the uterus and ultimately into the oviducts, also called fallopian tubes. Here he has a picture, which we don't have to go through. We've already discussed that. But not the prostate gland, prostatic urethra, the prostate gland. Let's go over here and see if I can pull one up. Prostate gland. Let's see here. The prostate gland surrounds the urethra. There's the prostate gland. It surrounds, but this can enlarge in a condition called benign prostatic hyperplasia, which would then cause, this would kind of, in many cases, close this off, which causes retention of urine from the bladder, which could be a problem. Now, that is a problem, and there are different drugs that can try to decrease that, and sometimes it involves surgery and so forth. We talk about erection, emission, and ejaculation, and I will start that one right after. We're going to close here and then start that.